Hello, everyone. My name is Michael Atwater. I am a junior biology and chemistry double major mathematics minor at Howard University. In the summer of 2019, I had the pleasure of researching at the Augusta University STAR summer program, and I had the pleasure of being in Dr. He's laboratory in the Department of Biochemistry and Cancer Biology. And my specific project was on the comparison of second generation CAR T cell effector function on hepatocellular carcinoma. So to start off, hepatocellular carcinoma, or HCC, is the most common type of primary liver cancer. It is the third leading cause of cancer deaths, and it is diagnosed in more than 500,000 people each year. However, there is no effective treatment, but immunotherapy has recently shown great potential as a cancer treatment. And a specific type of immunotherapy I am focusing on is chimeric antigen receptor, or CAR T cell therapy. And in this treatment, it applies cell engineering protocols to genetically modify a patient's own T cells so they can then go on and attack and kill cancer cells. So these CARs are engineered proteins that are composed of an extracellular fragment fused with a transmembrane component and then a costimulatory domain. So CAR T cell therapy specifically targets a cell surface protein, which is glypican or GPC3, which is overexpressed and only expressed in HCC tumor cells. And despite the benefits, CARs have not been FDA approved for solid tumors. Thus, it is necessary for us to carry out these preclinical studies and trials. And in this study, we generate a second generation CARs using an antibody produced in Dr. Hughes' laboratory, combined with three different costimulatory domains, ICOS, CD28, and 41BB. So for my hypothesis, I hypothesized that the choice of the costimulatory domain affects the properties of the CAR T cells. And as you can see in figure 1, it shows an example of a CAR receptor. You have, at the top, you have the single chain variable fragment, which is the part that recognizes the different tumor cells or the GPC3 protein. You have a transmembrane component, which anchors the CAR receptors into the T cell. And then we also have a different costimulatory domains, which is what I am testing. And the costimulatory domains actually send signals to the T cells and can have a different number of properties, such as affecting the viability and the functions of the T cells. So now for my materials and methods. In the beginning, we did the second generation CAR construction, which is we have a wide variety of protocols to actually make sure we have the correct DNA sequence for the CARs. We use PCRs, restriction enzyme digestion, ligation, transformation, different things to make sure we have the correct sequence for our different CARs. And after we had the correct DNA sequence, we did the lentivirus production, which is where we use transfection to actually produce viruses of the different CARs. And after we collected the viruses, we did transduction and we transduced our T cells with the viruses. So then now our T cells can actually express our different CAR receptors on the surface of the T cells. And then after that, we did immunostaining for CAR detection to see if the CARs were actually on the T cells. And after we did an LDH assay for cytotoxicity to see how they tested out and if they actually killed the cancer cells. So for my results, it shows in figure 2 that the bacterial transformation, and this is basically to see or to show that my bacteria actually transformed and accepted the different CAR DNA that I wanted them to have. And figure 3 shows the restriction enzyme digestion, and this is a gel electrophoresis image that shows that my specific CARs or the colonies had the correct length of DNA for the different CARs that I wanted them to have. And for figure 4, it shows like a summary of the CAR T cell transduction, and it shows a wide variety of properties that I am about to go into, such as the live cell percentage, the CAR expression, and also the percentage or the ratio between the CD4 T cells and the CD8 killer T cells. And as you can see, the first one is the non-transduced one, and that is set as the control or just a T cell, and these are the different costimulatory domains, so it shows the difference. And my next one is figure 5. It shows how the costimulatory domain affects the CAR T cell viability. And for viability, it's the percentage of live cells, and after the transduction, if the if it had an effect on keeping the T cells alive. And as you can see, the percentage of live cells increased from CD28 to ICOS and then to 4-1BB. And in figure 6, it shows how the costimulatory domain affects the stability of the CAR expression. And as you can see, going from left to right, in CD28, the CAR expression stabilized after day 10. So that means that it actually kept the different CAR receptors on it. And 4-1BB actually decreased as the CAR expression went on over time, and ICOS actually increased in their CAR expression over time. And figure 7 shows how the costimulatory domain affects the CD4 or CD8 ratio. And as you can see, the CD4 T cells are the helper T cells. And as you can see, with the different costimulatory domains, the percentage actually decreases over time for all of the costimulatory domains. But for the CD8 or the killer T cells, the percentage actually increases over time. And the one that increases the most is the CD28 that has the highest percentage, and that actually has a lower or the lowest percentage of the CD4 T cells. And my figure 8 shows how the costimulatory domain has no effect on cytotoxicity. So this was one test, and it shows that even though the different costimulatory domains, it has no different effect or varying effect on the killing of the tumor cells. So in summary, first we constructed and compared the second generation CARs using the different costimulatory domains. And after the cloning, we proceeded by the virus production and then T cell activation, transduction, and then we did staining with the fluorescent labeled antibodies. And we actually, in the end, we did the LDH cytotoxicity assay. And after that, we analyzed using flow cytometry. And in conclusion, the choice of the costimulatory domain affects the cell viability, CAR expression, and the CD4 CD8 ratio. And even though the initial data shows similar effector function, further studies need to be conducted to show if there actually is a difference in the killing of the T cells, and also other tests can be done about the cytokine production of our different T cells. These are my acknowledgements, and thank you.